Alright, so here's the deal. The sand, beautiful weather, summertime, getting tan, that's all over. This is you. You're about to leave that comfort zone of the summertime between M1 and M2 year and step forward into this world of unknown microbiology. So you and microbiology is ahead. What I want to say is that it can be very difficult to learn microbiology if we don't have a good structure to start on. So that's my goal with this video, is to help you figure out a way to structure learning all the different organisms instead of just learning them individually. So to start, we're going to look at gram-positive and gram-negative bacteria. That's our first step in differentiating. This is describing the cell wall, or a common way to describe the cell wall of different bacteria based on a test that you'll learn in lab. So gram-positive walls have a thick peptidoglycan layer which will retain the dye and end up looking blue or purplish. So gram-positive, blue, thick peptidoglycan. So here we have the gram-positive bacteria cell wall on the left side, cytoplasm being the inside of the cell, and then here's our lipid bilayer here. So both gram-positive and gram-negative, right, have the cytoplasm inside of the cell, as well as this inner membrane. Then there's this, so that's the plasma membrane. The next part is this peptidoglycan. As I said, gram-positive, thick peptidoglycan, and it's blue because it's going to retain kind of a bluish, purplish dye. So thick peptidoglycan and an inner membrane. Whereas the gram-negative bacteria have the inner membrane and then a really thin peptidoglycan. However, they have this inner membrane, but they also have an outer membrane. And this is going to be important. There's going to be a lot of key features in the gram-negative bacteria for the outer membrane. So how many membranes are in the gram-positive? Well, just one, right? We just have this inner membrane. And the peptidoglycan, how would we describe that? Well, it's really thick, right? So we have this thick peptidoglycan and just an inner membrane on gram-positive bacteria, and we're going to see them look purplish or bluish on gram stain. Now for the gram-negatives, we said there were two membranes, the inner membrane and the outer membrane. The outer membrane is going to be really important because we're going to see some features of it at the end here. The peptidoglycan, how do we describe this? Well, it's really thin, stuck in this kind of this periplasmic space between the inner and the outer membrane. So we have a thick peptidoglycan in the gram-positives and a thin one in the gram-negative. And the way I just think of it to remember is that the thick retains all this dye. So gram-positive, gram-negative. Another way you can think of it is that in order to squeeze a plus sign in between two bars, you need a lot of space. You need a lot of space for that peptidoglycan. Whereas the gram-negatives, to put a minus sign between two bars, you don't need a lot of space, and it kind of looks like it's adding an extra layer. So remember, the gram-negatives have that extra outer membrane, which is going to be a key feature, to have a lot of key features in it. So just think of the plus creating a big space between the bars, and that's going to represent the peptidoglycan. And then the minus looks like it adds like another layer, and it's going to have a thin peptidoglycan, can't squeeze a lot in there, and it's going to have two membranes, the inner membrane and that outer membrane. So the other thing to remember is that gram-negatives are negative, right? So no, anything that's red, stop, communist, stop signs, whatever you can think of. So red, negative, blue, positive. So moving forward to how to organize things. Flowcharts are going to be the key way to organizing bacteria. And we're going to start off by looking at the gram-positives. We're going to separate them first by shape. So we're going to look at bacillus, or rod-shaped, and cocci, or circle-shaped. So you'll hear bacillus referred to either bacilli or rods, and then cocci you'll hear just referred to as cocci. No one's going to call them, oh, the circle bacteria. But it's circular shape, so that's kind of what's going on there. We're just going to throw away these for now and focus in on the cocci to differentiate things. I think this is a very good place to start learning bacteria, so the gram-positive cocci. And this is where we're going to spend the rest of the time here. This we'll come back to later. There's kind of some important stuff to learn there too. The very first thing we do, right, is shape. So we determined that it was a cocci, so we did the gram stain. It looked bluish, and they were like little circles in there, right? So that's gram-positive cocci, and then we're going to do this catalase test to see if it's reacting or not reacting, and that's going to tell us whether we have staphylococcus, so these cocci that organize in little clusters, versus negative, where it's usually these cocci that organize in lines. So for the staphylococcus, first we do catalase, and we saw it was positive. So catalase positive, or staphylococcus, and determine what kind of staphylococcus we have. We're going to do a test called coagulase. So coagulase then, positive is going to be Staph aureus, negative is going to be the coagulase negative staph. The Streptococcus are going to be a bit more involved, and we're going to look at those now. So just to review, what shape was the Staph aureus? So Staph aureus was cocci, that's what we're focusing on, and it tend to be in clusters. So Staph aureus is cocci, and what shape was Streptococcus? Well, that was also cocci. That's what we're talking about. But look at this. Staphylococcus, Streptococcus. It's in the name. And then which of the two was catalase positive? Everything that's positive is going to stay on the left side of the chart here that we're describing. So catalase positive is going to go with staph. So Staphylococcus, always left side of the chart here is going to be positive. So I really want you to learn this chart, even though you may not know exactly what the tests are. We'll get into that farther on. So which test do we do on staph? So we saw that we had gram-positive cocci on the microscope, and then we want to determine whether it's staph or strep. We did catalase and we saw it's staph. So what test would we do next? The test we would do is coagulase to see if it's coagulase positive, coagulase negative staph. So which staph was coagulase positive? It's the notorious Staph aureus. So Staph aureus is both catalase and coagulase positive. That's going to be very, very important to know. So going to the Streptococcus, so these are the linear chained ones in general. We're going to separate these into three portions. So hemolysis, basically on a blood plate, we're going to see what kind of eating around, if you will, of the blood happens. It's going to be alpha, beta, and gamma, where alpha kind of has this like greenish hue around the bacterial colonies. Beta has like a complete clearing, so this is like way gone out there. And gamma is where it just looks like it didn't do anything to the blood itself. So this is a plate. What would plate look like? See here we have the greenish outline on the bacterial wall. The beta is completely clearing out here, and then the gamma doesn't look like it did anything to the blood plate. So alpha, green, and then our first test there is going to be the opticin. And positive or sensitive to opticin is going to be strep pneumo. So this is going to be a diplococcus with a capsule that's going to be very important for you to know. Definitely one of the more important ones to know. Then negative is going to be the viridins group. So these are things that are going to be in your teeth. So someone gets endocarditis after a dental procedure or something. So that's going to be opticin resistant viridins, the viridins group. Beta hemolysis is the complete clearing then. And then we're going to do this with grouping latex, or sometimes it's bacitracin can be done too. But if we do group A, see it reacting with group A, then we know it's strep pyogenes, or group A strep. Group B strep is going to be strep agalactiae. And the B you can think of for babies, because this is going to be very important for vaginal flora. So group B strep is going to be for babies, B for babies. Group A strep is kind of what we think of classically of what we think of as strep. So that's this group A strep. That's the one in the throat, the one without the cough, the one with the sore throat, and exudate coming out. Then the gamma one's going to be the PYR test. The positive is going to be Enterococcus, and the negative is going to be strep bovis. So the whole purpose of this video is to get you to memorize this chart of how to organize things, and then to have this outline and basically build in information. Like when you start learning about strep pneumo, start writing the information here, and you can have a visual mind map of where to go with everything, and it'll help you differentiate things on microbiology lab tests. So just in review, we have the gram-positive. That's the first thing we're going to do. We're going to gram stain it, look at the shape. They're little circles, the cocci. Then we do a catalase test. See if it's Staphylococcus or Streptococcus. If it's staph, we do coagulase. See if it's Staph aureus or a coagulase negative staph. If it's strep, what's the first thing we're going to do? We're going to look at what kind of hemolysis it has on the blood plate. If it's alpha, what are we going to do? The opticin. Like a little opticin disc drop there. If it's beta, then we're going to do either grouping latex or bacitracin sensitivity. Then if it's gamma, then do anything. Then we're going to do PYR and see if it's positive or negative there. So Staphylococcus tend to be in clusters. Streptococcus are kind of these linear chains. Strep pneumo is a good one to know that it's a diplococcus and it's capsulated. The other thing that I think is very important to know before you even start learning bacteria is kind of the common causes of meningitis. And for this, I want you to think of gram-bought home shopping network gel. And this is going to be a very weird mnemonic, very weird way to remember it, because I'm going to go backwards. And there's a very particular reason I'm doing this. So if we start with the gel part, we're going to think of G for group B strep, B for babies, remember, E for E. coli. Now this wasn't a gram-positive that we talked about, but it's something I want you to know. Listeria was a gram-positive, but it was one of those other category that I said we're going to ignore for now. But I really want you to know all of the bacteria that cause meningitis, common causes of meningitis, and the age groups that they fit in. So Graham bought Home Shopping Network gel. Starting off with the gel, we have Group B strep, E. coli, and Listeria. Next, Home Shopping Network, that's our second part of this puzzle, and this weird mnemonic going backwards. So gel being the first part, Home Shopping Network being the second part. The H is going to stand for H flu. And then this B, I like to think of it reminding me that it's type B H flu. The S is going to stand for strep pneumoniae. And then the N is going to stand for Neisseria meningitidis. So Home Shopping Network is the middle part here. And then Gram, like grandma, stands for gram-negative rods. Remember, 60 plus, like grandma. Okay, so grandma's buying this thing, and then 60 plus, what I mean here is that this is what's going to be a common cause of meningitis in older populations, so people over 60. Now, why did I go backwards with this mnemonic? Well, that's because this gel part is going to be common in the 0 to 6 month old. This next part, the Home Shopping Network, is going to be a little bit after that, so 6 months and older. H flu, we're going to think of 6 months to 6 years, and then the other guys, we're going to think even longer than that. So Neisseria meningitidis, we're going to go up to 60 years. And then gram-negative rods, we're going to think of over 60. The other thing I want you to clue in is that Listeria and strep pneumo come back, and they both also are a common cause in the over 60. So Graham bought Home Shopping Network. Remember, gel and the Home Shopping Network and Graham are going backwards in age. So gel being the youngest, Home Shopping Network being kind of the adult range, and then Graham and bringing back Listeria and strep pneumo for the over 60. And then just quickly going back to the cell wall to review, I think this is very important. Messed up this slide here, but this is just a preview, I guess. Then the lipid A, and then this core portion and O antigen. What this is showing you is LPS, lipopolysaccharide. I said that the outer membrane of the gram-negatives was very important. This is why. This is the endotoxin, or the LPS, lipopolysaccharide. A million names, just like everything else that you'll have to know. So I said it's right here. And this is very important because it's going to be embedded here. This lipid A, as you know, likes to hide away from water, so it's going to be embedded in the membrane. So if the bacteria is there, it's not going to cause any problems. But as soon as you start lysing and breaking down the bacteria, then your body responds to this lipid A portion. The rest of it isn't, your body isn't too responsive to, but the lipid A portion your body's going to react to, and you might go into shock. So that's kind of the danger there. Just to review, both have plasma membrane, both have the peptidoglycan, the thick one on the gram-positives, and then the gram-negatives also have this outer membrane too. Other thing to note is that there's a teichoic acid for the gram-positives. So I think the gram-negative LPS, endotoxin, is way more important to remember. But remember, you can remember the gram-positives also have a teichoic acid. So like I said, lipopolysaccharides, LPS, and here's the structure there. So O antigen, and then the lipid A. Lipid A being the thing that your body's going to react to violently in some cases. And then porin is another thing to know. That's a channel you can push things through, and that's in the outer membrane. So things that are specific for the gram-negative bacteria are going to be the lipopolysaccharide, the LPS, the endotoxin, that's all the same thing, the lipid A, that's all kind of the same, describing the same thing there, and the porin channels. And then for gram-positive is going to be the teichoic acid. So last review, remember, plus sign, squeezing in, need a thick peptidoglycan. The minus sign can just squeeze in very easily, and it looks like another extra membrane, and you don't need a very thick peptidoglycan to fit in there. So inner cytoplasmic membrane has that thick peptidoglycan that stains purple or blue, no outer membrane, no LPS, no porin channel, because all of these were going to be in the outer membrane. Here's the exception. Listeria monocytogenes does have LPS and endotoxin, but I would just throw that away for now while you're trying to get into learning bacteria. Where the gram-negatives, we said, also has an inner cytoplasmic membrane, thin peptidoglycan between the inner and the outer, but it does have that outer, and it's going to have the LPS, endotoxin, and the porin channel. So purple, blue, plus sign for gram-positive, and it's going to be thick peptidoglycan, pinkish red, and thin for the gram-negatives, and the outer membrane can be very reactive, or your body can react very badly to it. So lipid A, there we go. So that's all I have for you. The big thing here is learning that gram-positive chart. If I go back here, you know this chart, then you can start organizing your learning things. So you're going to start going through each bacteria one at a time, and it's going to seem very disconnected. But if you have this chart in a frame, then you can just put your notes underneath here. So strep pneumo, you start learning that it's a diplococcus and it's got a capsule. Strep pyogenes, you start thinking about, like people under 14 with sore throat that don't have a cough, that kind of thing. So on strep bovis, you start thinking about, someone has an infection, you start associating with colon cancer. But basically, this is your framework, and you have a way to organize all of your notes underneath here. And we'll do the same for gram negatives. Thanks for listening, guys.